Welcome to the deep dive. Our mission, as always, is to kind of cut through the noise and get you the core info you need quickly. Today, we're tackling a really hefty, super important chapter on microbiology lab basics. Think foundational techniques, controls, how to ID microbes, stuff every healthcare pro needs to know. We're basically giving you the practical bedrock.
And it is bedrock with a lot of history. I mean, to really get today's lab, you kind of have to look back. Back to the 1600s, the first microscope showing microbes existed. Then, the 1800s, Pasteur and Koch nailing down the germ theory, proving tiny organisms cause disease. It wasn't just bad luck. So all that work, verifying treatments, figuring out how diseases work, it absolutely requires control, a controlled environment.
Right, control. That's the theme today, isn't it? Because the stakes are, well, they're incredibly high. This isn't just book learning.
Exactly. The sources we looked at really hammer this home. This technical know-how is literally the front line. Think about verifying if a drug works or prepping for the next epidemic. These lab protocols, they're our shield against everything. Hospital infections, sure, but also really scary stuff like Bacillus anthrax, or toxins from Clostridium botulinum.
Yeah.
Getting the ID right and fast. It's literally a public health survival skill.
Okay, so if the lab is this critical, high-stakes place, how do scientists organize their approach? There's a framework, right? The 5 I's.
That's it, the 5 I's. It's a neat way to think about the workflow. Inoculation, incubation, isolation, inspection, and identification. We'll walk through those. But first, before we even think about growing anything, we have to talk about the absolute non-negotiable aseptic technique.
The cornerstone.
Absolutely the cornerstone. Aseptic technique is, well, it's this super strict set of rules. You do it under sterile conditions. And the whole point is to stop unwanted microbes, contaminants from getting in your sample or the lab or even onto you.
That sounds intense. Like creating a tiny sterile bubble in a world full of microbes. How does that actually work? How do you control something like, just the air?
It means being almost obsessive about the details.
Yeah.
You got to control air currents, so keep doors and windows shut. You need to manage fomites, too.
Fomites. Those are the inanimate objects, right? Benchtops, equipment.
Exactly. Things that can carry germs. And the sources give really concrete examples, like sterilizing your loops and needles, the little tools you use in a flame or incinerator, before you use them and after, every single time. And another key thing, flaming the mouth of a test tube or flask when you open it to transfer stuff. That quick blast of heat creates like an upward current. Helps keep airborne things from just falling in.
Okay, that paints a picture. So that strictness sets the stage for actually controlling the microbes. You mentioned 3 main levels: sterilization, disinfection.
Right, 3 key terms people often mix up. The difference is all about what you're killing. The absolute highest level is sterilization. This means destroying or removing everything, all microorganisms, and that includes the really tough ones like bacterial endospores, viruses, fungi, even prions. Total kill.
And the workhorse for that in the lab is the autoclave. What makes it so effective? It's not just heat, is it?
It's moist heat plus pressure. Think of it like a high-tech pressure cooker. It gets steam up to around 121 degrees Celsius, under maybe 1520 psi. That combination, the moist heat under pressure, forces heat deep into materials really fast. That's what gets those super resistant endospores.
Okay, so sterilization is the gold standard. Why have disinfection then? Is it just like a weaker version?
It's more about practicality. Sometimes you just can't sterilize something. Maybe heat would destroy it. Disinfection uses chemicals or maybe a physical process to kill most vegetative microbes and viruses. But, and this is the key difference, it doesn't reliably kill bacterial endospores. It lowers the numbers, but it's not total annihilation like sterilization.
And there's specific language here too, right? Disinfectants versus antiseptics.
Yep. Disinfectants are for surfaces, benchtops, floors, that kind of thing. Antiseptics are for living tissue, like using an alcohol wipe on skin before an injection. And it's worth noting, the sources say there's no single perfect disinfectant. They all have limitations. Maybe they're toxic or don't kill everything. It's always a balance.
Got it. And below disinfection.
Then you have basic cleaning techniques. Sanitization is more about mechanically removing microbes and debris to get things down to safe levels. Think restaurants, food industry using soaps, detergents. And degermination is specifically about reducing microbes on skin, usually involves scrubbing plus chemicals, like a surgeon scrubbing in before an operation.
Okay, environment controlled, levels of cleaning understood. Now we can actually start working with the microbes. Back to the 5 I's. First 2.
First 2 are inoculation and incubation. Inoculation is just introducing your sample, the inoculum, into whatever you're going to grow it in, the culture media. Then incubation is putting that inoculated container Somewhere with controlled temperature, usually somewhere between 20 and 40 degrees C, to let the microbes grow and multiply.
And that culture media, the food, it's not just one thing, is it?
Yeah.
It can be defined chemically.
Right. You've got chemically defined media where you know the exact formula, every single ingredient, down to the microgram. Useful for really specific research. But much more common, especially in clinical labs, is complex media. This has at least one ingredient, like peptone or beef extract or yeast extract, where you don't know the exact chemical composition. It varies a bit. It's generally richer, supports a wider range of microbes, and it's usually easier and cheaper to make.
Makes sense. And the media also comes in different physical forms: liquid, solid.
Yep. Three main states. Liquid media, we call them broths. Growth shows up as cloudiness, that's turbidity, or maybe sediment at the bottom, or sometimes a pellicle, which is like a film growing on the surface. Then there's semi-solid media. It's kind of like a soft jello, used mostly to see if bacteria are motile, if they can swim through it. And the most common is solid media, usually made solid with agar.
Agar, that's the stuff that makes it like jello, but it's special, right? Something about temperature.
Exactly. Agar is amazing stuff. It's a polysaccharide from seaweed. It melts at boiling, 100 degrees C. But, and this is the magic part, it doesn't solidify again until it cools way down to about 42 degrees C.
Okay, wait. Why is that 42C point so important?
Because 42 degrees is cool enough that you can pour it, even mix bacteria into it, without heat shocking or killing most of them. If it's solidified at, say, 60 or 70 C, you'd cook your sample trying to mix it in. That big temperature gap between melting and solidifying gives you a working window. You can melt it, cool it down to a safe temperature, add your bacteria, pour your plates or tubes, and let it set. It's incredibly practical.
Clever. Okay, beyond physical state, media can have specific jobs, right? Enriched, selective.
Right, functional types. Enriched media has extra goodies added, like blood, serum, special growth factors. It's for growing picky organisms, the ones that won't grow on just basic stuff. Blood agar is a classic example.
And selective, does that mean it selects for something?
Or selects against something. Selective media has ingredients that inhibit the growth of certain microbes while letting others grow just fine. The book uses mannitol salt agar, MSA. It's got a really high salt concentration, like 7.5% ACL. Most bacteria hate that much salt. But Staphylococcus species, they tolerate it. So MSA selects for staph.
Ah, okay. And differential media. That sounds like it shows differences.
Yeah. Exactly. Differential media lets different types of microbes growing on the same plate look different, usually based on some biochemical reaction, often shown by a color change. MacConkey agar is the go-to example here. It contains lactose and a pH indicator dye. If bacteria like E. coli ferment the lactose, they produce acid. The acid changes the dye color, so the E. coli colonies turn pink or red. But bacteria like Salmonella, which don't ferment lactose, they just grow without changing color, staying kind of off-white. So you can tell them apart just by looking. It's a visual clue.
Super useful. Which leads us perfectly into the third isolation, making sure you're looking at just one type of microbe, a pure culture.
Yes, absolutely critical. If you have a mix, you can't reliably test or identify anything. Isolation is all about separating individual bacterial cells on the agar surface so that each one grows up into its own little colony. And each colony ideally came from just one single cell, so it's genetically pure.
How do you do that separation? The main way is the streak plate, right?
That's the classic, the streak plate. You take your sample on a loop, and you basically spread it back and forth across one section of the agar plate. Then you sterilize the loop, cool it, touch it to that first section, and drag it into a second clean section. You repeat that maybe 3 or 4 times, streaking from the previous section into a new one. Each time you're dragging fewer and fewer bacteria. By the last section, you should have individual cells spaced far enough apart to form those nice isolated colonies.
Okay, I can visualize that. What about pour plates and spread plates?
Pour plate is different. You mix your bacteria sample into melted agar that's been cooled down to that safe temperature, like 45 C. Then you pour the whole mix into an empty petri dish and let it solidify. The colonies grow on the surface, but also inside the agar throughout its thickness. It's often used if you need to count the bacteria in a sample, and the spread plate is simpler. You put a small, measured amount of liquid sample onto the surface of an already solid agar plate, and then you use a sterile bent rod, like a little hockey stick, to spread it evenly all over the surface. Good for counting, too.
All right, pure culture achieved. Now we can really look at it. Fourth, eye inspection, which involves sticking them on a slide and staining.
Exactly. Inspection. And yeah, usually that means fixation first, killing the microbes and making them stick to the glass slide, often with heat or chemicals. Then staining, because bacteria are mostly clear and hard to see under a microscope without adding some color.
The most basic is just a simple stain.
Right. Simple stains use just one dye, usually a basic dye which has a positive charge. Bacterial cell surfaces are typically negatively charged, so the positive dye sticks to them, making them visible, shows you their basic shape, size, and how they're arranged clumps, chains, etc.
And the opposite is a negative stain.
Kinda, yeah.
Hmm.
Negative stains use acidic dyes, which have a negative charge. Since like charges repel, the dye doesn't stick to the negatively charged bacteria. Instead, it stains the background dark, so the bacteria appear as bright, clear spots against a dark field, like a halo effect. It's neat because you usually don't need to heat fix, which can distort delicate structures. Good for seeing capsules or really thin bacteria like Spirochetes.
But the really informative stains are the differential ones, right? Like the Gram stain.
Oh yeah, differential stains are huge. They use multiple dyes to tell different types of bacteria apart. And the Gram stain is the king. It's usually the first big test run on an unknown bacterium. It separates almost all bacteria into two big groups: gram-positive, which stain purple or blue, and gram-negatives, which stain pink or red. And it all comes down to their cell wall structure.
It's a four-step process, isn't it? Crystal violet first.
Yep. Step 1, flood the slide with crystal violet, the primary stain. Everything turns purple. Step 2, add grams iodine. This acts as a mordant. It doesn't really add color, but it complexes with the crystal violet inside the cells, forming these big crystal violet iodine or CVI complexes. Makes the dye harder to wash out.
Then the crucial step, the decolorizer. Usually alcohol.
Exactly. Step 3, rinse with alcohol or an alcohol acetone mix. This is the differential step. Here's what happens. Gram-positive bacteria have a really thick layer of peptidoglycan in their cell wall. The alcohol dehydrates this thick layer, shrinking the pores in the wall, and it traps those big CVI complexes inside, so they stay purple. But gram-negative bacteria have a much thinner peptidoglycan layer, and they have an outer membrane made of lipids. The alcohol dissolves this outer lipid membrane and easily washes the CVI complex out of the thin peptidoglycan layer, so they become colorless at this stage.
Ah, okay. And the last step.
Step 4, the counterstain, usually safranin, which is red. The gram-positives are already full of purple CVI, so they don't really pick up the safranin, they escape purple. But the now colorless gram-negatives readily soak up the safranin, so they turn pink or red. It's a really elegant system based entirely on that cell wall difference. And the sources note it works best on young cultures.
Fascinating. And there's another key differential stain, acid-fast.
Yes, the acid-fast stain. This one's specifically for bacteria that have waxy mycolic acid in their cell walls, which makes them resist gram staining. The main group here is Mycobacterium, the ones that cause tuberculosis and leprosy.
So that waxiness needs a special approach.
It does. You have to use a potent primary stain, carbofuchsin, and often apply heat, that's the Thiel-Nielsen method, or use a stronger formulation, Kinyon method, to drive that stain through the waxy layer. Once the red carbofuchsin's in it, that same waxy layer makes them resistant to decolorization by acid alcohol. So acid-fast bacteria hold on to the red stain, non-acid-fast bacteria get decolorized, and then pick up the blue counterstain. And then there are special stains designed to visualize specific structures that are otherwise hard to see, like tiny flagella or dormant endospores.
Okay, we've isolated, inspected with stains. Now the final I, identification. Putting a name to the bug, how's that done?
It's usually a combination of things. We look at morphology, shape, and arrangement we saw under the microscope. We look at cultural characteristics, how it grew on the agar plates or slants. And maybe most importantly, we test its physiological or biochemical characteristics. What enzymes does it have? What sugars can it ferment? What waste products does it make?
Cultural characteristics, what does that mean? Like how it looks growing?
Yeah, things like the texture, the color, the shape of the colony.
Yeah.
On slants, we use terms like filiform for thread-like growth or echinnulate for spiny edges, arborescent like a tree. Though these are descriptive, maybe less critical than biochemicals. A classic biochemical test mentioned is the gelatin stab. You stab bacteria into a tube of gelatin media. If the bacteria makes the enzyme gelatinase, it breaks down the gelatin protein. Since gelatin normally solidifies below about 28 degrees C, if the tube stays liquid even when chilled, you know the enzyme is present. The way it liquefies like crater-shaped or sac-like isn't as important as the fact that it did liquefy.
And those biochemical tests often come in sets, right? Like IMVC.
Right. The IMVC tests, that's indole, methyl red, Votus-Proskauer, and citrate, are a standard set, often used together, especially for identifying enteric bacteria, the gut bugs. They test for specific metabolic pathways. For example, methyl red and Votus-Proskauer test for different end products of glucose fermentation. Running the whole set gives you a pattern that helps distinguish closely related species, like different types of E. coli or Enterobacter. Another complex one mentioned is litmus milk reactions. You grow the bacteria in milk containing litmus indicator. It can tell you if it ferments lactose, digests milk proteins, produces gas. Lots of info from one tube based on color changes and clotting.
That sounds like it could take a while. Is there a faster way?
Definitely. Clinical labs rely heavily on rapid identification methods now. These are often packaged kits. Maybe you've seen things like the Tube 2 or API 20 strips. They're basically miniaturized panels containing maybe 15, 20 or more different biochemical tests in little wells. You inoculate the whole thing at once, incubate, and read the color changes. It massively speeds things up. Takes identification from potentially days down to maybe 4 to 24 hours. Some automated systems can even give results in like 6 hours. Crucial when time matters for treatment.
But even faster and more precise is looking at the genetics, right? Molecular analysis.
Absolutely. That's really the cutting edge and often the most definitive, molecular or genetic analysis. Especially given how incredibly diverse microbes are, this is super important for bugs that are slow growing or maybe can't even be grown in culture easily, or ones that look identical biochemically but are actually different species.
How does it work, basically, cutting up DNA?
One common way involves using restriction enzymes. Think of them as tiny molecular scissors that cut DNA only as specific recognition sequences. So if you treat DNA from a specific bacterium with a specific enzyme, it will always get cut into fragments of predictable lengths. Then you separate these fragments by size using gel electrophoresis. The fragments migrate through a gel based on size when you apply an electric current, creating a unique pattern of bands. It's like a DNA fingerprint for that species.
Very cool. And DNA probes.
DNA probes are another powerful tool. These are short, specially synthesized pieces of DNA or RNA that are designed to match and bind to a specific target sequence in the microbe's DNA or RNA. You label the probe somehow, often with a fluorescent tag. If the target sequence is present in your sample, the probe sticks to it, and you get a signal. Really useful for detecting specific pathogens quickly, even if they're hard to culture, like certain mycobacterium species. And today, sequencing the DNA of ribosomal RNA genes is extremely common. Those genes are essential, highly conserved within a species, but different between species, making them great targets for really accurate identification.
Wow, okay. So pulling it all together, if someone's listening, trying to get the big picture of the microlab from this chapter, what are the main takeaways?
I'd say number one, everything hinges on strict aseptic technique. Can't do anything reliably without it. Number two, we use a whole arsenal of culture media, carefully chosen, enriched, selective, differential, to grow what we need and see what it's doing. Three, the gram stain is still that fundamental first look, dividing the bacterial world based on cell wall structure. It's quick, informative. And four, identification is moving rapidly towards molecular analysis, DNA fingerprinting, probes, sequencing, because it offers unparalleled speed and specificity, especially for tricky organisms.
That definitely brings us to a powerful final thought. We hear about threats, bioterrorism agents, new pandemics, food contamination, and the response always seems to involve testing. The ability to develop things like handheld PCR devices for detecting anthrax spores, or those rapid tests for food pathogens or viruses. It all rests on this foundational lab science, just accelerated by molecular tools. It really feels like the lab is the bedrock of global health security.
It truly is. That translation from basic bench techniques to rapid field deployable diagnostics is happening constantly, and it's vital.
Absolutely. Well, this has been incredibly insightful. Thank you so much for breaking down these essential techniques for us. And a big thank you to you, our listener, for tuning in to this deep dive. From the whole Last Minute Lecture team, we really appreciate you being here.